my Tech Futures talk. My name's Dr. Donna Whelan and I work here at Trobe University in Bendigo. And today what I'm going to be talking about is the next generation of fluorescence microscopes. So all of my research really pulls together physics and chemistry and biology. It's very interdisciplinary, but it all kind of coalesces to give us some really fantastic images inside cells using really advanced microscopy. So to let you know where the talk is going, I will give you a little bit of background on me because hopefully that gives you the context to understand how I ended up doing the research that I'm doing. I'll then talk a little bit about this idea of single molecule microscopes and single molecule biology because it's probably not something that you've heard about before. It's a fairly new area of science. And then talk about how that comes together in the form of what we call super resolution microscopy. That will then give us a segue into what I actually use these amazing techniques to study. And at the very end, what I'm going to walk you through is how we actually built the microscope that I use because we were very fortunate to be funded locally to build a one-of-a-kind setup here in Bendigo, and it's now been up and running for about a year. So in terms of what I'm hoping to convey in this talk and what I'm hoping you learn from this talk is just how interdisciplinary science can get. I'm always surprised thinking back to my high school experience, my undergrad experience, even my PhD, you kind of get boxed in as being a chemist or a biologist or a physicist. But when it actually comes to research, lots and lots of people straddle multiple fields. And in particular, my research really does come across all of that, as well as things like computer science and engineering and maths. So hopefully that gives you an idea of what interdisciplinary science looks like, because I don't think it's something that is very well represented in high school or undergrad. I also want to give you a little bit of insight into the amazing research that's going on here at La Trobe Bendigo. Obviously, I'm just one small part of a very big team of researchers, but hopefully this gives you a taste of the kind of things we can do here in Bendigo that really there's no one else in the world doing the research that we're doing. And then finally, just again, that idea of bottom-up single molecule biology. It's a really new take and it's an interesting way of thinking about the world around us. So who am I? As I said, my name's Donna Whelan. I'm an Australian. I grew up down in Melbourne in the southeastern suburbs, but in a very strange kind of coincidence, my dad's whole family grew up and lived in Bendigo. So throughout my childhood and my teenage years, I was coming up here to Bendigo 4 or 5 times a year and visiting my grandfather in his old house, which is still there on High Street in Golden Square. So growing up down in the southeastern suburbs, I was lucky enough to get accepted into Monash University, where I did a Bachelor of Arts, Bachelor of Science double degree, because I didn't really know what I wanted to be when I grew up. At the end of high school, I just knew that I was interested in learning and definitely wanted to go to university. So through that 4-year course, I was able to study all sorts of sciences. So chemistry and physics were my main ones, and then all sorts of arts, so things like history and English, and ended up getting a major in philosophy as well. After that, I took a year off because I still didn't know what I wanted to be when I grew up, and I traveled and I worked full time. And then I realized I was actually missing being in a research lab, being in a wet lab at a wet bench doing chemistry. So I came back and I did a PhD in chemistry. So that all finished up back in 2015. And then I was lucky again in that I was able to secure myself a job in New York City, working at the New York University School of Medicine. So after a PhD, you typically try and do a few years of postdoctoral research before you settle down into a permanent job. And I was very pleased to be able to go to New York for a few years and really move out of chemistry and into a much more medical, biomedical kind of research sphere. Did that for a few years and then came back and got a job here at La Trobe in Bendigo. So that started in March of 2018 in the Department of Pharmacy and Biomedical Sciences. And it was my first kind of independent job setting up my own lab. That's gone really well for the last 2 years. And now it's successfully transitioned onto a Discovery Early Career Research Award, which is a Australian Research Council early career support kind of startup package. And it gives you money and it gives you a position that's government funded. And that's secured my position here until 2022. And then I'm lined up to just become a permanent member of faculty, teaching and doing research. So that's my whole story, and hopefully you'll see from the techniques I use, the techniques I develop, and the research that I do, that interdisciplinary kind of trying lots of different things out, trying to fit them together, has really flavoured the research that I do. So what do I mean by single molecule biology? As I said, it's not a very familiar term, and it's something that has only really come around in the last decade or two. And I like to think of it as the bottom-up approach to doing biology. And that's kind of borrowing some terminology from information technology. So when you're thinking about a really complicated computer system, you often talk about understanding it either from a top-down or a bottom-up kind of approach. And simply a top-down approach takes a very complicated system and breaks it down into its parts to try and understand it. And that's how we've traditionally done most biology. Whether you're studying an ecosystem or an organism or even a single cell, you start with a very complicated system and then start breaking it down in order to try and understand it. The bottom-up approach is to try and understand the most fundamental building blocks of a system and then start building that system from those fundamental building blocks. So in biology, our fundamental building blocks are single molecules. So all of my research really revolves around trying to understand humans, trying to understand disease and dysfunction and cells, one molecule at a time. That has its own set of challenges, and there's a reason that most people haven't really seen it as a viable way of doing research. And that's simply down to numbers. So I always like to pose this question to people when I'm describing what I do. Which of these is going to be the biggest number? The number of molecules in one adult human, the number of grains of sand on the planet, or the number of stars in the universe? And hopefully what you already are aware of is that there's lots of sand on planet Earth and there's lots of stars in the universe. To give you an idea of the numbers we're playing with, the number of cells in an average human body is about 37 trillion. So that's quite a big number. And then the number of molecules in an average cell is about 2 trillion. So if you want to know how many molecules there are in a human, you just have to times those two numbers together. But how does it actually stack up compared to sand? So the estimated number of grains of sand on planet Earth is 7.5 quintillion, which I'm sure you can comprehend is an enormous number. It pales quite poorly when you compare it to the number of stars in the universe, which is 1 septillion. So there are far, far more stars in the universe than there are grains of sand on planet Earth. If you times those two numbers at the top together though, you get 74 septillion. So there are actually 74 times as many molecules in one human as there are stars in the universe. So we're talking about really, really big numbers, really numbers that we can't really comprehend. We certainly can't process this amount of data. You know, we don't have the computing power to put this many bits of information into a machine. So studying biology bottom up remains enormously challenging. You know, it's not the intuitive way to try and understand cells or diseases or humans, but it has some really powerful advantages. So we are trying to do it anyway. And the way that we're doing it is in the super resolution microscopy technique. So super resolution microscopy is a form of fluorescence microscopy. It's really about harnessing special kinds of light, because once you can specifically visualize one type of light and ignore all the rest, you start to be able to filter out the molecules that you're interested in. So the special type of light that we look at is a form of luminescence. So luminescence is the emission of light by a substance, by a molecule, without heat. So there's lots of things when you heat them up, they'll start to glow, and there's heat in that. There's lots of things that will react, produce heat and light, and then there are things that just produce light. And this is a really beautiful example of bioluminescence, where you've got bacteria in the water that actually have a biochemical reaction that emits this bright blue light. So super resolution microscopy is a form of fluorescence microscopy. Fluorescence is a form of luminescence. Other types of luminescence include this kind of breaking of chemical bonds, and just breaking those bonds spits out these little bits of light. And then the other form of luminescence, which is very closely related to fluorescence, is phosphorescence. So all of the things that you buy that glow in the dark, glow sticks, these stars you stick on the ceiling, they're all forms of phosphorescence. Fluorescence sits in the visible spectrum. So we do get to play with all of these really beautiful visible wavelengths. There's lots of science that uses infrared and ultraviolet and X-rays, but you can't see them. So you have to detect them some other way. Because we're working in visible light, so any of these colors here, we can actually just look down a microscope and visibly see that. So fluorescence, again, because of that specificity, because you can pick it up and ignore all the other forms of light simply by turning the room lights off, if you like, has been a really powerful tool for biologists because they've gotten very, very good at painting their targets. So this is just a Google search of fluorescence in biology, and you can see all of the different structures and organisms and cells that have been visualized using fluorescence in the past, using microscopy. So it's already a cornerstone technique for biology in a very top-down approach. This is still looking at really big, complicated systems, and any one of these images has millions, if not billions, of fluorescent molecules in it. In terms of single molecule fluorescence, in the late 1980s, single molecules were observed fluorescently for the first time. So this is just an example of that. This is just some glass, which doesn't fluoresce, so it looks black. And then on it, we've put some single molecules. So each one of those little blobs is one molecule that we're seeing. And that was first achieved, about 40 years ago, 30, 40 years ago. And these days, it's very common. If you have a good enough microscope, you can see it by eye. You can just look straight down the microscope and say, that's one molecule that I'm seeing. So there's a huge problem that goes along with all of this. And it was really the defining limitation of fluorescence microscopy up until about a decade ago. And that's this thing called diffraction. So diffraction describes the way that light behaves. And it's effectively a blurring effect because the laws of physics dictate that the size of visible light is much bigger than the underlying size of the molecule. And that's just how it is. If you're looking at something red, the wavelengths that you're seeing are 600 nanometers big. That's still a tiny, tiny number, but it's still much bigger than a molecule. So what you're looking at here, excuse the chemistry, is one of the most famous and most widely used fluorescent molecules. And it's actually on the bigger side for fluorescent molecules. And this molecule is about 2 or 3 nanometers across. So if we draw that to kind of scale and say that little dot there is our molecule, it will actually look about this big when you look at it on a microscope. It'll emit enough light that you can easily detect it, easily get this nice round shape, but it's 200 or 300 nanometers big, whereas the underlying molecule is only 2 or 3 nanometers. So it's about 100 times as big as the molecule. And what that means is that when you've got lots of molecules clustered together, everything just starts to get blurry over that 250 to 300 nanometer scale. Worth remembering though, these emissions that are 2 to 300 nanometers big, you could still line up thousands of these across the world. We're still working in incredibly small scales. However, cells are incredibly small and the structures that make them up are also incredibly small. So we had to come up with a way around this, and that's really what super resolution microscopy does. So this is just one small subsection of a cell, and we've painted up a thing called microtubules. So you can kind of think of these as being the bones of a cell. So we've painted them with this beautiful green fluorescent paint, and we can then go onto a microscope and take a picture. But what you might already be thinking is that's a bit blurry, and it is. This is as good as it gets because that light is just blurring out, overlapping with each other. You know, this image has millions of fluorophores all overlapping and emitting at the same time. Now, it's a very different story if you can actually look at just one fluorophore. So this is now just 5 of these fluorescent molecules, and each one of them is spitting out these very nicely circular blobs of light. If we know that we're only looking at one molecule and we get this very nice circular blob, what we can actually do is mathematically solve where the molecule is. And then all of a sudden we know within that 200 to 300 nanometer blur where the underlying molecule is located. And that's really the trick that goes into super resolution microscopy, because you take your 2 million fluorophores and you just start to look at them one by one. So again, that bottom-up approach of, okay, let's go one molecule by one molecule by one molecule. What does that look like when you put it all together? So again, you've got your blurry fluorescence image on the left, and you've got our super resolution, much clearer, much sharper, much more information image on the right. And to get there, we have to do a lot of physics, a lot of chemistry, because we have to manipulate these molecules so that they do this blinking. So it's exactly the same area of the cell, but we've changed how it behaves so that these molecules sometimes are spitting out light and then they go dark. And by having them go dark, we can look at all of these molecules one at a time. So we'll take 10, 20,000 different images of different molecules emitting. And then for every single one of those blobs, we can actually work out to within a few nanometers where the molecule is. And then when we paint that into space, knowing those localization coordinates and just dotting, we get that much clearer image that's on the right. So we see everything about 100 times more clearly. You go into 3D, it's about 1,000 times more clearly. You're seeing so much more detail. And this is detail that's real, that's about the inside of the cell that you're looking at. So what do we actually use that for? So I'm just going to talk about 2 research topics that I'm currently working on or have worked on in the past, because I think they're 2 of the most interesting. And everything about these has really been published already. So if you want to know more, it's all out there online. So the first one is DNA damage and repair. So DNA is the molecule that underlies all life on Earth. You know, it codes for who we are. It codes for inheritance. So your DNA is a combination of your mother and father's. And if you have kids, you will pass on some of your DNA to them. But it codes for everything from hair color to foot size, height, and then things like behavior, you know, how you react to certain things. It's all kind of encoded into DNA. So it's a really important molecule, and it's really important that we don't damage it. Because when you damage it, you change your genetic code, and that will either lead to cell death, which can be bad, or mutation, which can also be really, really bad. The thing to remember here, though, is there's lots of ways that damage our DNA, which are really easy to repair. So looking across this double-stranded double helix, you can see a number of different kinds of damage. So something like a single strand break is when one side of that double helix, just the backbone of it breaks apart. Also get things like mismatches or damaged bases or intrastrand crosslinks. And that's just affecting the genetic code of one side of your double helix of DNA. And the whole reason that we have 2 strands of DNA, that we have double-stranded DNA, not single-stranded DNA, is because that other strand carries an identical copy of the information. So as long as you have one strand intact, you can use it to repair the other strand. The real problem here isn't really all of this damage that's only on one side, it's the double-strand breaks. To put in context how much more dangerous they are, you have about 200,000 single-strand breaks and damages per cell per day. So all of those damaged bases, single-strand breaks, interstrand crosslinks, all up, it's about 200,000 per cell per day. And as I said, any kind of change to your genetic code can lead to mutation, cell death, disease. These really don't cause very many diseases in humans. We don't think that they're responsible for the mutations and the cell death that do play really big roles in human disease. We think it's the 10 double-strand breaks that actually trigger all of our cancers. So it's these that really underlie human disease and dysfunction in a lot of ways. And that includes, as I said, all of your cancers have to start with some sort of DNA damage. We think that things like motor neuron disease and dementia have some form of neurodegeneration, which potentially links back to cell death that's caused by DNA damage. And then things like immune system dysfunction is probably related damage. Things like genetic diseases, they're all forms of DNA damage. So we really want to work out how these double-strand breaks occur, how we can stop them from occurring, how we can repair them better. And that's really what a lot of my research is focused on. So this is an example of an image of one cell, just the nucleus. So the whole cell is actually quite a bit bigger than this kind of round blob. But we just focus on the nucleus because that's where all of your DNA is. And what you can see is in the top left, you've got this blurry image, which is epifluorescence, which is just your normal kind of fluorescence microscopy. And then in the bottom right, you've got the super resolution image. So it's the same cell, we've just imaged it with and without single molecule detection. And what you can probably see straight away is how much more information is in that bottom right. And that's because in the cell nucleus, this is something that's, you know, only about 5,000–6,000 nanometers in diameter. So it's quite small already, but you then have to take into account that you've got billions of base pairs of DNA. You've got about 2 meters of DNA in there. And so it's really highly compacted. As well as that, you've got lots and lots of proteins that are going around maintaining the DNA, replicating the DNA, repairing all of this damage. So it's an incredibly dense environment. And the denser the environment, the more that blurriness has an effect. Because if it's so dense that everything is just blurring together, as it kind of is in the top left, there's really no straightforward way to start pulling out meaningful information. And you can see that in the two zoom in sections over on the right, where we're kind of seeing a little bit of blue and a little bit of yellow, and these are proteins that we're interested in. We want to see how these proteins are interacting and interacting with the DNA, but you really can't say much structurally about what's going on. In comparison, those zoom ins on the bottom and the super resolution image on the bottom right, you start to see really clear, crisp structures. And what we're looking at here is in the magenta, you're looking at active replicating DNA. So we're just focusing in on DNA that's being copied. And then we're actually causing some of that copying to go wrong and generate some of these double strand breaks. And then we can see this pair of proteins that we think play big roles in repairing DNA double strand breaks. We can see how they're co-localizing with these active replication sites. We can start to pull apart how they're interacting with each other. And that's really an incredibly powerful approach to studying DNA damage. This is a lot of work that was developed while I was in New York. So the lab in New York and my lab here at La Trobe Bendigo, we're really the only ones that have combined kind of biochemistry and molecular biology and the super resolution microscopy to be running these experiments. So what does that actually tell us about the repair response? So this was work in my postdoc. And what we were able to do is you've got this very complicated kind of schematic on the left, which shows a lot of the proteins involved in DNA double strand break repair. And this has all been solved using things like biochemistry, molecular biology techniques, a lot of bench techniques, and very little in-cell observation, because the in-cell observation really requires the super resolution. So the first thing that we did with the super resolution was go back and look at this pathway and see if we've got the same sequence of events. And hopefully you can tell by this heat map where the white squares are. That's when we saw really high co-localization between these proteins and the DNA damage sites. And you can kind of see we've put them in the same order that that the literature has them in, and you can see that as time progresses from left to right, you do have these early responders and then middle responders and then late responders coming at different times. But the really nice thing about this data is every single one of those white boxes or light blue boxes contains 100 images, and each of those images contains hundreds, if not thousands, of double strand break sites. And we can go back and look at every single one of those and start to pull out spatial information, 3 and 4 color information, so we can see how the proteins interact with each other. So that's kind of just the flavor of that research. And we've got so many different avenues and collaborations looking at different proteins, looking at different pathways, looking at different ways to cause the damage, to repair the damage, that the technique is definitely proving really powerful. So just one little example of that. So this is much newer data. We're looking at a protein called NBS1. So this is a protein that it's very well known that it's related to some pretty severe genetic diseases. And recently it's also been implicated in antiviral responses. So it's quite an interesting protein, and it's very well known that it goes to DNA double strand breaks. It is recruited to these DNA double strand breaks, and then it will interact with the DNA, and it has some role there in helping repair it. But there was also some kind of speculative data that showed it was getting recruited to what we call nucleoli, which are these loose regions of DNA within the nucleus that are responsible for generating proteins for further protein synthesis. So they're a really specific kind of subcompartment within the nucleus. And it's still not very clear why this protein might be going there in response to DNA damage, but nobody had ever been able to see it. And what I hope that was completely expected. But what you can see in the bottom zoom in, and hopefully you can start to kind of see in that main image, is there's a few of these denser magenta areas. And what we can see straight away is you don't get these little points of NBS1, you get kind of a dusting of it. And we only see that in the nucleoli. We don't see it anywhere else in the cell nucleus. And so this really strongly supports that idea that for some reason NBS1 is going to these nucleoli. It also explains why no one's been able to see it before, because when you get these really strong points of light versus something that's quite dispersed on a normal microscope, the really strong points are the only thing that you'll pick up. Whereas here, bottom-up biology, single molecule biology. We detect every single molecule that's there and we can treat them molecularly. So hopefully that's a flavour of the DNA damage work that my lab does. Something else that's quite interesting and quite topical was work that I did a few years ago in collaboration with Greg Mosley and Toby Bell, who were both at Monash University, and they were interested in the rabies virus. So we're very lucky in Australia that we have absolutely no rabies, to the point where we couldn't even do live rabies research in Australia. We had to get some collaborators in Japan to work with the live virus, kill it, and then send it over. So we don't have rabies here. We have related viruses in some of our bats, but internationally rabies is still quite a serious problem because it still causes about 60,000 human deaths per year. And the really, I guess, scary thing about rabies is that if you contract rabies, we have really good treatments, which is why if you're ever in a country where there is rabies and you get bitten by a dog or a bat, you immediately go to hospital and get the treatment. Because if you wait until you start to feel sick, you're pretty much guaranteed to die. It's 100% fatal once it's symptomatic in humans. So I find that really quite terrifying. But we don't have it in Australia. So we were interested in it because our collaborators had been trying to do conventional fluorescence microscopy, and they weren't getting any straightforward answers to go with all of the other biological research that they were doing. What they were interested in were these microtubules. So again, the cell bones. And as soon as we put it on the microscope, we kind of had our answer for them. So this is these microtubules in a human cell that is kind of infected, if you like, with a specific part of the virus, this thing we call P3. So it's got all of this P3 in it, and we know that the P3 is quite deadly to cells. And as soon as we looked at the microtubules, we saw all of these really thick cords, and they're about kind of 200 to 300 nanometers big, which, if you remember, is how big they would always look if you're on a normal microscope, because everything looks 200 to 300 nanometers big. Even if it's one molecule wide, it's going to look 300 nanometers big. But we knew from all of our other work that microtubules look about 60, 70 nanometers big when you look at them using single molecule imaging. So we were a little bit surprised that we were seeing these microtubules that seemed so much bigger than they should have been. And our collaborators kind of went, well, how do we know that your technique's working? So the first thing that we had was, if you can see on the right-hand side of this image, there's another cell there, and we knew that that cell wasn't infected. And you can see coming out of it lots of very skinny microtubules. So we were pretty hopeful. And then what we were able to do is we were able to get a version of the virus that wasn't as fatal, that wasn't anywhere near as dangerous. And when we looked at microtubules in that cell, as you can see here, they went back to being, for the most part, single microtubules. So you can see in the zoom ins, they're kind of 60, 70, 80, maybe 90, but that one, you can tell that there's kind of 2 overlapping microtubules going into it. So in this cell and in all the cells that we looked at that were infected with this much less fatal version of rabies, we only saw genuine single microtubules. So that was a really quick answer. I mean, it still took us years, but it was a really straightforward, here's the power of the technique, because they'd been looking at diffraction limited images for a while and they weren't able to really tell this thickness difference. What was nice about that was when they started collaborating with us, they had quite a lot of information about these viruses. So the one on the left is your wild type, and that's the one that has really high rates of fatality, really. The one second in, we call an attenuated version because it's really not pathological at all. And then we had these 2-point mutants, which were kind of somewhere in the middle because we mutated the virus to try and make it more or less safe. So what they already knew was that the wild type rabies really antagonized the immune system. So it really activated the immune system in humans, in mice, and that can actually be quite dangerous. Really high levels of immune antagonism can be more dangerous than the virus itself. The non-pathological one didn't do so much antagonism of the immune system, and then our 2 mutants were somewhere in the middle. They'd also been able to find that this protein associated with microtubules. So they weren't able to tell the thickness differences, but they could see that sometimes, in the wild type, it would colocalize and overlap with the microtubules, whereas in the non-pathological, it really didn't associate with them at all. And what we were able to add in to all of this data was another kind of clue as to how it all fit together. So in the case of the wild type, which you know has high immune antagonism, has high P3MT association, we also found really high microtubule bundling. And then in the non-pathological, we also found really low microtubule bundling. So all of these things seem to be following the same trend, even in the point mutants. And at the end of the day, what really matters is the fatality rates of these things. And we were able to show that ability to bundle up the microtubules. We still don't really know why it's doing it, but the fact that it can bundle them up seems to tie in with it then being incredibly fatal. So this is all in mouse studies. And in the wild type case, it was found to be 100% fatal. The non-pathological version didn't kill any of the mice. They barely even got sick. And then our 2 point mutants, once again, kind of somewhere in the middle. So this was a really nice application of the single molecule microscopy into a much bigger, really interdisciplinary biology project. So that's just a little taste of the research that's kind of been done and is ongoing in my lab. And now, just for the last kind of 5 or 10 minutes, I want to actually show you the microscope and how we built it and what makes it so powerful. Because I think I've talked a lot about biology. Certainly most of my research is biology focused, but the actual microscope itself relies so much on physics and chemistry and then in engineering just to get the whole thing together. So it's all up and running. These are some really stunning pictures we took of the microscope. You can see that there's quite a lot of electronics going on. There's lots of moving parts. At the front end, it kind of looks like a microscope that you might see on TV, but at the back end, it's quite a complicated mess. And you can see in the bottom right image there, there's really bright lines of light are our laser excitation sources. So this is something that a normal microscope wouldn't have, but we need them. And they are very, very bright. They're a little bit dangerous. So it's fortunate that I have a very nice lab to put them in, but very, very stunning to actually see every day I go to work. And once again, the real reason this got built, and it got built here in Bendigo, is because of a donation through the Bendigo Tertiary Education Anniversary Foundation, which paid for everything we needed to build one of these. And it is one of a kind in the world. It's completely bespoke. You'll see from all of the bits and pieces that go into it, we had to work with quite a few companies and order lots of kind of custom bits and pieces to build this exactly the way I wanted it to do the research that I do. So what are all the bits and pieces that go into something like this? The first thing you need is a really top-end camera. So these cameras run kind of $50,000, and you need that because you want to be able to detect every single light particle you can to get enough signal from one molecule. So I said detecting molecules is reasonably straightforward these days. It absolutely is if you can get your hands on one of these very, very nice cameras. And you know, they're $50,000. They're not particularly big. This would be about the size of about that big. So quite compact, and it just sits on the side of the microscope. So if we look back here, that's the camera just kind of sitting in there on the side of the microscope. So we got one of those, and it's running beautifully. We need these high power lasers, because as much as we can use the camera to detect as much light coming out, one of the things that dictates how much light comes out is how much light you can put in. So you need really, really high power continuous wave lasers, and we got 5 to cover all of the visible spectrum. So we've got a 640 nanometer, which is a red, a 561, which is a yellow, a 532, which is a green, a 488, which is a blue, and a 405, which is violet. So they're all lined up on the table, they all run all the time and look a bit like a rainbow. To stabilize all of this, we had to get a very heavy table. It's about a ton. So we were very fortunate that we were able to get a second-hand one from St. Vincent's down in Melbourne, but then of course we had to transport it up to Bendigo. And that top of the table is a solid slab, so there's no way to kind of dismantle it. We had to make sure we were going to be able to get it into the room. So it all took a little bit of planning. We worked with Olympus to customize the actual frame of the microscope. So that front end user-friendly kind of interface and a lot of the electronics as well. And then we bought literally tens of thousands of what we would call optics and optomechanics. So all of these little bits of metal, you screw them together. It's kind of like Lego. So as I said, I'm a biologist most of the time, but sometimes it does just come back to being a bit of a tinkerer. And then we also had to get a pretty high power computer because the data generation on this thing is a lot. We can easily do about 0.5 terabyte a day. So this all kind of kicked off in 2018. It was months of planning, months of ordering, negotiating, making sure that I hadn't forgotten anything and making sure everything would fit together. And then on the 6th of November, back in 2018, we got our first laser. So this got delivered from Hong Kong and it's our green laser, and it just had to sit in my office until everything else started arriving. What you can probably tell from that picture, that's just a normal Sharpie. Even the lasers are really quite small and compact. So mid-November, we got all of that optical Lego. So it was 5 massive boxes that again, just kind of sat in the corner of my office because I couldn't really start doing anything with them. But we did unbox them and look through it all. It was a bit like Christmas. 13th of December, just before real Christmas, we knew that we were getting the table. And again, it's 1 ton, so we had to be really confident that where we put it was going to work because there's nowhere to move that after the fact. You have to get in specialist equipment to lift it. People just can't lift a ton, no matter how many people you go around that table. So I spent some time down there really mapping out where it was going to go and then put it in and got it in the right spot. We haven't had to move it yet. And then by the 17th of December, we had all 5 lasers. So again, that's the green one in the middle. It's the biggest one and it's still quite small. Some of these are worth $20,000, $25,000 and put out hundreds of milliwatts, which is really quite intense light. But they're just boxed up into these tiny little boxes that fit in the palm of your hand. And then on the 18th of December, exactly a week before Christmas, the actual microscope frame started rolling in. So these are all custom bits and pieces from Olympus. And of course they turned up the week before Christmas, just before I got kicked out of the lab and had to go and do real Christmas. I was a little bit sad that I didn't get to stay and actually start building. I did unbox it though, but as soon as I could get back in on the 2nd of January, we started putting it all together. So getting it on the table where I wanted it, planning it all out. Same day, this was always part of the plan, but we didn't really want to commit to it until we saw the dimensions of everything else. Went to Bunnings and started buying up all of the industrial shelving that we would need, and then spent a day building it around the table. So this is really to hold a lot of our cabling and our electronics. More shelves and the computers started to go in. And about 2 weeks later, so this is 2 weeks to really get the lasers on the table, get everything connected, get everything talking to each other. By the 15th of January, we had over 1 watt of laser belting around the table and looking quite cool as well. A few more weeks and we started putting all of this Lego in place on the table. So these are all very, very high quality metal and glass. And this is really just used to control where the laser goes on the table, using things like mirrors and dichroic glass. Once that was all in, we could turn the lasers on and actually get them bouncing around the table and hopefully see here that you've got your bank of 5 different lasers lined up here. One of them is yellow, but iPhones just don't detect yellow light very well for some reason. And they all come up and they bounce off all of these different optics. And then this is actually the back of the microscope. And you can see once they're all combined, it is a really nice white light that's going into the back of your microscope. And then a few days later, we actually had the Trobe students in the lab looking at things on the microscope. So once everything was planned and ordered, it was a pretty quick build. And since then, yeah, we've been doing all sorts of exciting research on it and letting the undergrads play with it as well, which is quite cool. So these are some of the images that they got. Again, looking just at cells. The green here is those cell bones again. We're looking at DNA in blue, really letting them experience some of my research as well. And about a week later, we pulled it all together and using the lasers, we're actually able to see the first single molecules. And that's kind of the end of that story. So thank you all so much for listening to my Tech Futures talk. A special thank you once again to the Bendigo Tertiary Education Anniversary Foundation, without whom this just wouldn't have been built. Thanks to the Trobe Institute for Molecular Science, who employed me in the beginning, and now the Australia Research Council, who are have put me on one of these DECRA fellowships.
Okay, Donna. So I know that a lot of students will probably be watching this recording and video, and I guess a question that would be something that they would ask is, what were your interests in high school?
Yeah, it's a good question. And I did do chemistry. I was really pushed to do chemistry. And I actually did like the accelerated program where you do year 11 chemistry in year 10, and then year 12 and year 11. And then by year 12, I was doing first year chemistry at Monash. But I wouldn't say at that point I was very interested in it. I was good at it, I guess, but I was still feeling pretty rebellious and wasn't pleased that I was being made to do extra work. I do distinctly remember in year 12 having a bit of a crisis about this push for me to go into science, because I did really love the humanities subjects. And my year 12 English teacher and homeroom teacher at the time actually wrote in my yearbook that I needed to start realizing that I could do both, that I could do humanities and science. And that was one of the real reasons that I chose to do a double degree. Again, there was a lot of kind of pressure to just go and do a straight science degree or a biomed degree or medicine. But I really wanted to pursue the humanities a little bit. And even though I've ended up in science, my skill set is very much derived from my interest and my experience in the humanities. So even though I don't really use philosophy on a daily basis, not many people do, the ability to think critically and to argue persuasively, those are some of the skills that I got from pursuing the humanities that have made me a much, much better scientist.
Yeah, I think that's quite fascinating for you to sort of mention that. And I know in high school, a lot of students are sort of pushed one way or the other. So it's quite interesting to hear that throughout high school you were pushed in both directions. And I know sometimes we categorize ourselves as either a left brain or a right brain, but your interest sort of worked with both. So I think it definitely makes sense why it has worked for you as a scientist and following those passions as well.
Yeah, look, it's advice that I would give anyone who isn't 100% sure that they want to do X. Keep trying other things, because I wasn't sure that I wanted to be in science until, I took a full year off at the end of a 4-year degree. And I'm happy to own up to that, because it meant that when I came back, I was really passionate and felt sure of myself. Whereas looking around, there are a lot of people who are even starting PhDs. And you think, why are you doing a PhD in chemistry or biology or whatever? And they just say, oh, well, that's what I've been doing for the last 7 or 8 years. I started in high school and I've just kept doing it. And I think that doesn't usually work out very well. So you have to want to do it, not just be able to do it.
And that kind of flows on, I suppose, Donna, to the next question, which was, a lot of school leavers or school students at the moment might be able to really relate to not knowing what they exactly want to do. And I think you've already answered the question, if you're unsure, there's definitely options out there to keep your interests there at university. I suppose the next question is, did you have a break between your undergrad and doing your PhD, or did you go straight to doing your PhD, and why?
Yeah, so I took a year off between undergrad and my honours year. I really wanted to have a gap year at the end of high school, but it wasn't really an option. So I just went straight into university. But by the end of that 4 years, and I really spent the first 2 years focusing on getting my science degree, and then the last 2 years really branching out into the humanities. At the end of that 4 years, I wasn't sure that I wanted to do either of them. And I was in a privileged enough position that I you know, had a pretty stable full-time job by that point. I had a lot of friends and family that could support me if I needed it. So I thought I'm going to take a break and just kind of have a think. You know, I was managing Subway stores, so it wasn't like it was a career that I was pursuing, but it was enough to kind of give me something to do. I traveled. I spent a couple of months overseas that year. And then, yeah, realized I really missed being in a lab environment. And I also, you know, I missed a lot of the kind of essay writing and critical thinking that had gone through the arts degree. And I thought, well, that sounds like research. So honours is the year that you can usually tack on to the end of a 3-year undergraduate degree. And it's a much more dedicated research year than the rest of your degree. So I came back and did that, and that was fantastic. So I straight away enrolled into PhD.
I think it's quite interesting how it's not a conventional path. I know when you hear the word gap year, a lot of students will probably think that it is something that happens straight after high school, but I think it's interesting that you mentioned how critical that was in your journey to have that gap year after you've experienced a little bit of study and then you've gone and done some things that aren't necessarily in that field. And I think it's really important that even though it was a gap year, it wasn't necessarily something after high school, but it still helped shape your journey. And yeah, I find that super fascinating that that helped you reiterate what you were interested in and sort of help you gain some clarity into your future as well. So I know that you mentioned that you did some study in New York City. So I guess how was your experience doing your postdoctorate in New York City?
New York was fantastic. I had been to New York a few times before I got the job, just for holiday. And I love it as a city. It's, you know, insanely busy, insanely bustling. I'm a huge musical theatre fan, so that's kind of the place to be. So it was really fortuitous that there was a job that came along in New York, on Manhattan, you know, 40-minute walk to Times Square. So just from a kind of life experience point of view, it was a really fantastic opportunity. In terms of the science, it was a big shake-up to go from a chemistry department in an Australian university to a, you know, working hospital that has a school of medicine, you know, on Manhattan, incredibly busy, you know, a for-profit university, much more kind of competitive and I guess internationally recognised as one of the top places to be. So that had its own challenges to kind of get used to that difference in environment and differences in expectations. And I had to learn a lot of biology because even at the end of my PhD, I was still mostly a chemist, you know, with more physics than biology at that point. And to go into a department of biomolecular pharmacology was what it was called. I had to really go back and learn lots of undergrad biology. So that was an interesting challenge. And then I guess the third aspect of moving to New York was kind of the potential isolation. So, you know, I had no network there. I didn't have any friends there. I moved on my own. So that was a bit of a challenge to get used to that, to build up new networks. But fortunately, I now have friends and colleagues over there, and I worked really well with my professor over there. So we still collaborate, and as part of my research, I have funding to go back pretty much every year to keep that kind of collaboration going. So it was a fantastic experience, and it really set me up for this research that I now want to do here in Australia.
That's really awesome, Donna. And I saw that there was some awesome opportunities for undergrad students to be mentored or use the microscope that's just been built. Do you have any advice that you give the students when they come in and see the microscope, or would never have thought that they'd actually get a chance to use a piece of equipment like this?
I think a lot of it for me is just about exposing students to research and to the fact that it's interdisciplinary and it's collaborative, and that science as a career, as a researcher, is actually quite different to science as an undergrad or a high schooler. Because I don't think that ever got communicated to me very well during my undergrad. So I do like to bring the students in, and I do a little bit of lecturing now where I can fit it in, and we do experiments on the microscope that the students can take part in, and just getting them in there and showing them what an active research lab looks like, and getting them to realise that when they look down the microscope and see something, they're the first people that ever saw it. You know, it hasn't been imaged before somewhere else. This is our special sample, our special experiments. You know, if they're in an undergrad lab class, they're doing a lab practical that has been done 1,000,000 times by 1,000,000 different people, and they know what the right answer is, or at least the people around them know what the right answer is. Whereas in a research lab, every time you do anything, it's going to be a bit new, and you don't know what the answer is going to be. So really trying to instill that that's what they can potentially get to do at the end of an undergrad is probably my main kind of push.
I think that's quite a unique perspective to look at it. Obviously, you've gained experience in the field and you know exactly how you like to feel during those moments. So to sort of give that opportunity to students as well is quite fantastic. And I know that to hear about the endless possibilities that there is in research, especially in this field, is quite fascinating. Sort of is quite motivating to hear as well. So I guess you being a great mentor to these students leads us to the next question is, have you had any great mentors throughout your research or throughout your studies that have really influenced you as a scientist?
Yeah, I mean, you have to have great supporters to succeed in science. I don't think you can get through it without good support. And I think I've seen both extremely good mentors and not so great. So to focus on the extremely good ones, one of my PhD supervisors, Toby Bell, so we worked together on that rabies research, and we still work together almost weekly. We'll touch base because we co-supervise students and we collaborate on a number of projects, and we're developing, you know, it used to be very much mentorship because he was a PhD supervisor, but we're now developing this kind of collaborative relationship where he's really the expert in chemistry and physics and I'm becoming the expert in biology. And so we're quite a good team, but certainly through my PhD, through even my postdoc when I was in New York, I would routinely go to him for advice because he'd obviously been through all of the problems I was having. He also postdoced internationally. And he was a really good mentor because he didn't have any of that bias where he was trying to get something back for himself. He just saw it as part of his kind of job, even though we no longer had or have a professional relationship. He saw it as his job to try and make me into a better scientist. So he's still a very, very good mentor. And then I think also I did a lot of work at the Australian Synchrotron during my PhD, and there was a support scientist over there called Keith Bambury, and he was incredibly instrumental through honours of my PhD on mentoring me in the kind of politics side of science. And it's terrible to realise that there is a political side of science, but you have to be able to navigate a lot of different personalities and a lot of passion. And he was really fantastic for advice on that.
That sounds awesome, Donna. And I know that I've had some really great mentors in the past that have framed my engineering career. And so I can really relate to that as well. And so I think at the Tech School, we call it a bit of a moonshot idea that we try to get the students to think about was with this microscope, and we can see already the amazing capabilities that it has. But if there's one thing that you could do, it doesn't have to be one thing, could be a contribution to many things. But if there was one thing in the future that you could achieve with the microscope, what would it be and why?
There's two different ways that I want to answer this, because on a very practical perspective, you know, it's really hard to predict where science is going to go in a research lab because it's just too many variables. So from that practical perspective, the real ambition with this microscope is to get as many people using it as possible, because it's a very powerful piece of instrumentation, and it's always constantly giving us new data, new ideas, new insights into, you know, I have dozens of collaborations through different viruses, different bacteria, different human diseases, animal diseases. And the more people that are actually using it and coming in and generating data, analyzing data, the better chance we have of having one of these moonshots happen, because I don't think many planned moonshots happen. I think they happen when you're just doing lots of good science. So that's the kind of practical side. I guess the thing that I would really love to be able to do, and it's kind of something that we have thought about a lot, is really be able to go in and start looking at one DNA event. So at the moment we look at a whole cell and we look at tens or hundreds of events at once, and we have to kind of statistically analyze them, and then there's a little bit of guesswork that goes into really pulling out the data that we've got. And what would be fantastic, and I think is potentially possible, is to be able to go in and look at one event. So for example, it's many people's moonshot to look at a single replication fork, you know, a single site where DNA is replicating, and actually be able to tell the difference between the leading and the lagging strand. So the two different sides of DNA replication. Now these things happen, you know, within a few nanometers of each other. So using a microscope didn't seem possible, but we're now starting to think maybe we can come up with a way to do it. So I'd love to try and visualize that. And then the other thing that I've been trying to get going, I just need time and students to work on it, is gene editing. So there's a technique called CRISPR-Cas that has revolutionized gene editing, both in the lab, and we think eventually it's going to be out, you know, editing the genomes of organisms and potentially humans. But again, we've never been able to actually just go in and see how that editing is happening. And so that's something that is less of a moonshot, because I think we're actually going to get it to work in the next few years. But to be able to see these individual editing events where the DNA is cut up and then you change the sequence and there's lots of different proteins, if we can see it, we can actually start to better control it and understand it. So that's kind of the ambitions for the next 5 to 10 years, I guess.
Donna, it has been an absolute honour to listen to you today, and I know that so many people, whether they have a science background or not, will find today's conversation so fascinating. So a massive thank you from the Bendigo Tech School for sharing your knowledge and your research. I know that it's going to reach a lot of people, and I think it's going to inspire a lot of people too. So thank you so much.
Thanks, Donna.